Okay, good morning and welcome everybody to WESTalk, Water and Environment Student Talks. WESTalk bring together leaders on sustainable water and environmental research from around the globe and students interested in making a difference. Later on, I'm going to show you the lineup that it shows leaders and experts in the field that we invited since January, and a map that it shows the audiences attended from around the globe for our WESTalk. With that, first I would like to talk about the WESTalk Conference 2021, which is a fully virtual conference based in UBC. It's everything about water, and abstracts are due April 15. Please do attend and submit your abstracts for poster presentations and talks, and there are going to be a lot of cool prizes as far as I know. And WESTalks, it's an initiative by IC Impacts and UBC Future Water, which both of them are based in UBC. Here is the organizing committee: Abhishek, Fuhar, Jaskaran, Carl, Freya, and myself. Most of us are from UBC, and Jaskaran joined us from University of Guelph. Here's the map that I talked about. This shows the global reach of WESTalk, which actually makes us really happy to see how diverse our audiences are. And here is the lineup, sorry, from January 14. Today's April 1st. Speaker is Dr. Jane Fowler from Simon Fraser University. She's going to talk about drinking water biofilters, which is one of my favorite topics. I recently defended my PhD in this drinking water biofilters. And before giving an introduction about today's speaker, I would like to reiterate, we actually talk about this every time, that you can find the previous WESTalk on YouTube channel of IC Impact. And a brief introduction about today's speaker, Dr. Jane Fowler, who is an assistant prof of environmental biotechnology at Simon Fraser University. The goals of her research group are to develop and improve sustainable biotechnologies for drinking water, wastewater, and contaminant treatment using open microbial community, and to understand the impact of microbial communities in engineered systems with a view towards ecosystem and public health. She completed postdoctoral training at Technical University of Denmark, Department of Environmental Engineering, where she was awarded a Marie Curie Fellow and was awarded a PhD from the University of Calgary. With that, I would like to hand it over to Jane, and please feel free to take it away. And everybody, if you have any question, please put your question in the chat box, and Jaskaran will moderate the question at the end of the presentation talk. I'm going to stop sharing here. Jane, it's all yours.
All right, can you hear me and see the slide? Okay, good. All right, so yeah, first I'd like to thank the organizers for inviting me today to this really cool seminar series that you guys have developed. I also want to thank everyone that's attended to listen to the talk. So today I want to talk about drinking water biofilters. Mostly I'm going to talk about some of the work that I did during my postdoc at the Technical University of Denmark, since I just set up my group here at Simon Fraser University. And we're still in the early stages, setting everything up in the lab and getting things going. So here's an outline of the talk for today. So first I'll give you an overview of biofiltration, the different types of technologies that are used for drinking water treatment by biofiltration, and then give you some examples of different drinking water treatment processes that are used in different locations that apply biofiltration technology. And then I'll look at this project that I was involved in, which was a survey of Danish drinking water treatment plants based on rapid sand filters. And then I'll wrap up with some thoughts about perspectives on the drinking water microbiome and its importance in the drinking water distribution system and potentially in public health. All right, so biological drinking water filtration technology is a pretty ancient technology. There's references to it in ancient Rome and Egypt. But today there's three major processes that are still used to produce large scale drinking water on a large scale. So sort of the probably most simple one and maybe the oldest one is bank filtration, where the sediments on in the riverbeds are used as a filter in order to produce clean drinking water. One that's been also widely used, especially across Europe in the 1900s, is the slow sand filter. So that you can see a picture of a slow sand filtration plant here. This picture is from 1927 in England. And you can see these huge filters that take up a lot of land, which is kind of the drawback of these slow sand filters, is the amount of space that they take up, but they're a very effective way to produce clean water from surface water or groundwater. And so the sort of most high-tech version of these biofiltration technologies is the rapid sand filter. And here you can see probably what's the most high-tech rapid sand filter I've ever seen is this. This is a pressurized rapid sand filter in the city of Aarhus. And you can see they've installed all these sampling ports down the sides so that you can take samples of both water and filter material throughout the depths of the filter. So these rapid sand filters are an improvement in terms of the size of the slow sand filters. And each of these different technologies has different aspects that give them benefits and drawbacks. But the basics of a biofilter are pretty much the same no matter which one you're looking at. So mostly these biofilters will consist of a filter bed that can be of varying depths with gravel or coarse rocks at the bottom, and then they'll be filled with sand or some other type of filter material throughout the filter. These might be single media filters, or they can be dual or multimedia filters. And common contents are things like sand, anthracite, granular activated carbon, and then some specialized filtration materials that can help with improving removal of specific contaminants. And so I was talking about slow sand filters and rapid sand filters. So the main difference between these different types of filters is the speed at which the water flows through them. So in a slow sand filter, you have a much slower velocity, so a longer HRT. So you have a longer time for the water purification to occur. And then in rapid sand filters, the velocity of the water is much faster, so shorter HRT and a less contact time for the treatment to occur. And the main processes occurring in drinking water biofilters, there can be chemical, physical, and biological treatments. So filtration is important by straining, flocculation, and sedimentation. Also absorption can occur. This is especially important with these specialized filtration materials. So for example, GAC can be used to absorb organic carbon from the water, and then specialized materials like zero valent iron can be used in order to absorb things like arsenic and uranium and other heavy metals. And the last process that's occurring in these biofilters, and which is my most favorite, is biodegradation. And so this occurs because this very high surface area that you get on the surface of the sand or the filtration material allows for the growth of a lot of to a rich bacterial community on the surface of these sand grains, and these organisms are involved in the removal of a lot of the contaminants in these filters as well. And when you're using something like GAC, actually, where you're absorbing pollutants onto the surface of these filter materials, that can actually allow for enhanced biodegradation of complex pollutants, because by basically immobilizing these pollutants, you're giving the bacteria more time to remove them. All right, so let's take a look at some of the different processes that are using drinking water, or that are using drinking water biofiltration in their drinking water production technology. So first, I want to look at the one that is closest to us here in Metro Vancouver. So Metro Vancouver serves a lot of the lower mainland's water needs. The Seymour Capilano filtration plant uses biofilters, and it's the largest treatment plant in the area. And it can filter up to 1.8 billion liters of water per day. So the water at Metro Vancouver comes from, at the Seymour Capilano plant, comes from Capilano Lake and Seymour Lake. So these are, it's obviously surface water that's being treated. And the process looks a bit like this. So the water comes in from the lakes, and then it goes through a coagulation, flocculation, and sedimentation process, which helps to remove a lot of the larger particles that are present in the water. And then they have 24 2-meter deep biofilters at the plant, and the filter material inside consists of a mixture of sand and anthracite. So the water goes through these biofilters, and then this filtered water is treated with lime in order to reduce the corrosivity of the water. And then it's disinfected using chlorine. So many of you might be able to taste the chlorine. Maybe you can't. It sort of depends on how sensitive your taste buds are and how used you are to chlorinated water. So something sort of similar, but also a little bit different, is the way that drinking water is treated in Zurich. So Zurich also uses surface water, just like Vancouver. So they're using water from Lake Zurich. But in this case, they're not actually disinfecting the water with a residual disinfectant. So the water is not distributed with a residual disinfectant like chlorine or chloramine. So they do have some disinfectant steps. They use ozonation. They use a process where they have ozone and then filtration in series. So here you can see they pump up the water from the lake. It's then ozonated. This removes a lot of the bacteria, and it also oxidizes a lot of the organic material. And then it goes first through a rapid sand filter into a second ozonation step, and then through a back filter, which is a granular activated carbon filter with biological activity in it. And then finally, it goes through a slow sand filtration process. And as I said, the main difference is that there's no chlorination at the end. And so you can see the treatment process is actually quite a bit more intensive. And this is because essentially so that they are able to distribute this water without a residual disinfectant. So the idea is that they're producing water that's biologically stable, which means that there is insufficient nutrients in the water that's being distributed for bacterial regrowth to occur in the distribution system. All right, and so then lastly, I want to look at the drinking water treatment process that I'm most familiar with personally. This is the one in Denmark. So this is the way that drinking water is produced across the entire country. All of the drinking water in Denmark comes from groundwater, and it is also biologically stable. So it's not disinfected before distribution. In fact, it's not actually disinfected at all through the entire treatment process. So the groundwater is pumped up into the treatment plant, then it goes through an aeration step in order for the biofiltration to be aerobic. The aeration process can vary from sort of very low tech to high tech. So here we have a very low tech example of aeration processes in Denmark where water just flows down these aeration steps and in that way takes on oxygen. Then the water goes through a rapid sand filter. This might be one rapid sand filter, or sometimes there are two rapid sand filters in series. And then this water is directly distributed to the consumer with no disinfection. So this is actually, it's quite interesting to me. So this is the treatment process in Denmark that is mandated by federal law. And I've been told that actually the reason that this is mandated by federal law is to encourage responsible agricultural practices to reduce the use of pesticides. So they're actually pushing farmers to reduce their pesticide use by minimizing the treatment of drinking water that can be performed. All right, so I said this was the one that I'm the most familiar with, and this slide will sort of explain why. So While I was a postdoc at the Technical University of Denmark, I went to a lot of these treatment plants. So here I'm showing you the location of 13 different treatment plants that we looked at for the survey project. We looked at 8 treatment plants that were in the Copenhagen region, and then 3 others that were elsewhere in the island of Zealand. So those was here. We looked at one on the island of Fyn here, and then one in Jutland. That was actually this pressurized filter that I had the picture of in the first or one of the early slides. And so what does the water actually look like? So this is showing the major contaminants that were present in the influent water coming in, so in the groundwater. The main contaminants that we see in this water are ammonium, and then iron and manganese, and there's also some methane and sulfide. And so you can see here the amount of these contaminants in influent varies a lot by location. The main thing, so this is these green lines in the bottom. These are actually the regulatory limits. So you can see some of these waters are actually already meeting the regulatory limits with no treatment whatsoever. Many of them are not, but actually all of these treatment plants do meet the regulatory limits before the water is distributed. So they're all performing well. In addition to ammonium and metals, we also see a bit of methane as well as sulfide, typically due to the stripping that would occur during the aeration process. They're in very, very low concentration when the water actually enters the filter, and they tend to be below the detection limit by the time they get to the start of the filter. And so we wanted to look at the microbial communities that were growing in these rapid sand filters. We knew that, so if we go back to this slide, we knew that the ammonium is mainly removed by biological processes. Nitrification is the main way that ammonium is removed, so it's converted to nitrate via that process. The metals, we expected that iron would be removed mostly chemically due to exposure to just direct oxidation by oxygen when it comes into contact with air. Manganese, we thought it could be biological or chemical. And then methane and sulfide, the concentrations were so low, we weren't sure what to expect with respect to that. But we wanted to look at these microbial communities, and so we applied a few different methods to study them. So first, we wanted to look at who is there. So we applied amplicon sequencing. So basically what we did is we took filter material from these filters, we extracted the DNA, we did a PCR amplification of the 16S rRNA gene, which allowed us to identify the organisms that were there and determine something a bit about their evolutionary relationships to each other. We also wanted to know how many organisms were actually present. So we did a quantitative PCR. So we can do that to just actually measure the gene copy numbers of the 16S RNA gene and as well as other marker genes. And then we also did some metagenomic sequencing. So that can tell us what can these organisms do, what are they actually capable of, what do they have the gene catalog for in order to actually do metabolically. And we were able to actually recover 132 genomes from these 13 different rapid sand filters. So these will tell us a lot about the organisms that are present there and what they're capable of. Lastly, we wanted to, well, at one point we needed to figure out which organisms were capable of doing what. So we applied a method called stable isotope probing. And this is a method that uses 13C labeled or stably labeled, in this case we use 13C labeled bicarbonate in order to look at which organisms are actually using the substrates in the filters. So this is a way that you can label organisms biomolecules. So the biomolecules in these organisms that are using these substrates will take up this isotope label. And if you can get sufficient labeling of these organisms, you can actually separate the labeled biomolecules and unlabeled biomolecules using ultracentrifugation techniques. And in that way, you can identify the organisms that are actually performing the metabolism you're interested in. In this case, we were identifying the autotrophic nitrifiers that were incorporating 13C labeled bicarbonate while they were oxidizing ammonium, as well as ammonium and nitrite. The last method we used was some old-fashioned microbial isolation and characterization. So this just involved enriching cells directly from the water or extracting cells from the sand material and then growing them up in liquid media or in plates, and then characterizing these individuals. All right, so what did we find? So this image here is a picture of the amplicon sequencing results that we found. So basically it's showing you the identity of all of the organisms that we found at the phylum level. And so you can see there's a lot of different organisms present in these sand filters, but there's also quite a lot of similarities between the communities that we see. And this was not particularly surprising to us because the chemical profiles of the incoming groundwater were also quite similar, and this is what's feeding these biofilters. So we weren't too surprised to see that there was a quite similar profile between the different sand filters. The qPCR that we did told us that the total numbers of organisms present in these sand filters varied quite a lot. So from around 5 times 10 to the 7 to 1 times 10 to the 10 organisms per gram of sand. Most of them were actually around 10 to the 9 organisms per gram of sand. There's quite a lot of bacteria in these filters. This was measured just at the top of the filter. There's going to be a lot less organisms as you go through the depths of the filter. There was a very high microbial diversity. We had several thousand different organisms present in each filter. But mostly what we see from these pictures is that these proteobacteria in pink and the Nitrospira in blue are by far the most abundant organisms. The metagenomic data showed us that we have a high functional potential, which I'll tell you about a bit more later as we go on. Okay, so this phylum level picture doesn't really give you a good idea of the actual functions of these organisms. So we did an analysis to look at just the core community. So that's the organisms that are shared between all of these filters. And we looked at them at the genus level, which would give us more of an insight into potentially into their functions. And so what we found was that when we looked a bit deeper, there were 9 genera of organisms that made up between 57 and 96% of all of these communities. And you can see these organisms here. And they only made up 10.6% of the actual organisms that we see, but they made up a very large proportion of the total diversity in the filters. And so we can look a bit deeper into these and see something about their functional potential. So these are the main functions that we were looking for, biological functions that we were looking for based on the influent, the influent coming into the filters. And so nitrification, we thought that was going to be the most important process biologically. There's Nitrospira and Nitrosomonas here. Nitrosomonas is a typical ammonia oxidizing bacteria, and Nitrospira is a typical nitrite oxidizing bacteria. Metal oxidation, hypomicrobiome, and pedomicrobiome are typical metal oxidizers. We saw this methyloglobulus, which can oxidize methane and methyl compounds. And then the organics oxidation, a lot of these organisms aren't very well characterized, but we knew that these 4 taxa that were also in the core community were probably oxidizing organic carbon. And so the organic carbon input into these filters isn't very much, but if you think about the biofilters, the organisms themselves, so we're talking about 10 to 9 cells per gram, are actually a great source of organic carbon. So that's probably an important food source for these organisms. All right, so let's talk a little bit more about nitrification. So this is where the story got really interesting for us. So nitrification, in case you're not too familiar with it, it's the biological oxidation of ammonium to nitrate. And normally this is the process that's been known for over 100 years. And it's a process that's split into 2 different organisms typically. So there's these ammonia oxidizing bacteria and archaea. These are organisms like Nitrosomonas, which we saw in the core community, Nitrosospira, Talmarchiota. And these organisms, so they oxidize ammonium, they produce nitrite. The key enzyme here is ammonium monooxygenase, and they get quite a lot of energy that they can conserve from this process. The second group of bacteria are the nitrite oxidizing bacteria. Again, in the core community, we saw Nitrospira were quite abundant in the filters. There's also Nitrobacter and Nitrotoga that are well-known nitrite oxidizing bacteria. And so these organisms don't get quite the same amount of energy, but still it's quite okay. But we would expect to see a lot more of these ammonia oxidizing bacteria than nitrate oxidizing bacteria. The key gene we look for here as a marker gene is the nitrite oxidoreductase, or NXR. And so what we didn't know when we started this process, when we started this whole project, was that there's actually a special group of organisms, the Collomox nitrospira, select lineage 2 nitrospira, that are capable of carrying out this entire pathway. So this was a process that we discovered in these filters, and it was really exciting because I think it was about 3 or 4 different groups, 4 different groups found this process all at the same time. So there was us and then 3 other groups that found this, that discovered this metabolism all sort of at the same time. And it turns out to be quite abundant in groundwater treatment filters. So these Collomox nitrospira oxidize ammonium all the way to nitrate, and they contain both the AMO and the NXR genes. And so you can imagine when we first looked at this picture, which is showing all of the nitrifying bacteria that are present in these filters, we were totally flummoxed over why these nitrospira were so abundant when they were only thought to be nitrate oxidizing bacteria at the time. We would have expected to see a lot more AOB, but these nitrospira were dominant. And it was later when we looked at our metagenomic sequencing that we found I guess the key to this was that these were also ammonia oxidizing nitrospira. And so, like I said, we were very confused, but we thought maybe something was wrong. There was some crazy bias in our 16S RNA gene sequencing, but it turned out that, I mean, our qPCR results agreed with everything. The nitrospira were the most dominant. Nitrosomonas and Talmarchiota were always present, but they were in very low abundance. And the same when we looked at the nitrospira. So again, our 16S RNA Illumina results, so that's the sequencing. It agreed quite well with our qPCR results, looking at the 16S RNA gene and the NXRB gene. So as I said, we found in the metagenomes that there were also these nitrite oxidizing nitrospira. So that allowed us to create a new qPCR assay to look for the AMOA gene specific to nitrospira. And the quantification of that really agrees well, again, with what we're seeing with our Illumina sequencing and with our other qPCR. So a lot of these nitrospira that we see in these filters are actually ammonia oxidizing or complete ammonia oxidizing nitrospira that are carrying out the nitrification pathway completely from ammonium to nitrate. And so because, you know, it's sort of this big paradigm that the nitrification pathway is divided, we had to really show that these nitrospira were active ammonia oxidizers in the filters. So what we did is we did signal isotope probing. So as I said before, we were looking at the organisms that were incorporating labeled ammonium or labeled bicarbonate during the oxidation of ammonium and nitrite. And so this picture, it's a bit complicated, but it just shows showing the different lineages of nitrospira. This is lineage 2, where these Collomox organisms are found. And if you see a filled in bubble, that means that it's a Collomox nitrospira. So we saw both Collomox and nitrite oxidizing nitrospira in the filters. And the nitrospira in the filters took up the greatest percentage of 13C label in the ammonium fed columns. Nitrosomonas also took up 13C label, but it was somewhat less than the nitrospira. And the nitrospira were actually the only nitrate oxidizers that we were able to identify in the drinking water filters. And they were the only ones that were taking up bicarbonate, labeled bicarbonate. So that was a really exciting finding, to find this totally novel physiology in these drinking water biofilters. All right, so we also wanted to look at the other biological functions happening in these filters. And one thing that surprised us was the relative abundance of methane oxidizers. So you can see here, this is showing the abundance of methane oxidizing bacteria in the filters. So remember, methane, there's hardly any methane. It's undetectable most of the time going into the filters. But what we found was there was up to like 25% of the population was actually consisting of methane oxidizing bacteria. And most of them were this type 1 methane oxidizer. These are typical methane oxidizers that are found in fresh water and in groundwater sediments. So that wasn't really surprising to find that type of methane oxidizer. So there's been some methane oxidizers found that are actually believed to be able to basically persist in the presence of atmospheric methane conditions, which is quite incredible that they're able to support their growth and survival just based on these extremely low concentrations of methane. So we also looked for methane oxidation pathways in the metagenomes. We used a marker gene for the methanol dehydrogenase, and we found it was present in all of the filters to varying abundances, but it was present everywhere. So similarly, we didn't expect to see very many sulfide oxidizers. When we looked for them, we actually, again, found them in almost all of the filters. We found especially two different species of well-known sulfide oxidizers, the Sulfuretalia and the Thiobacillus. And the interesting thing about these was that, at least from the Illumina data, the 16S RNA sequencing data, seemed like they were mutually exclusive. So we would only find one or the other, but we always found one of them in the filters. And again, when we looked at our metagenome data, we found there's two major pathways for sulfide oxidation. One is DSR based and one is STOX based. And we found both of these pathways in all of the filters. So again, there are sulfide oxidizers present, even though the sulfide levels are almost undetectable. And then lastly, I had some students do a project looking for manganese oxidizers. So manganese, manganese is one that's interesting because it's typically not believed to be linked to energy conservation. So manganese oxidation is believed to have something more to do with detoxification generally, for the most part. So there's lots of organisms that can oxidize manganese, but it's not necessarily linked to their growth. So we were able to, the students, Aska, Frederick, and Lasse, went to a bunch of different drinking water treatment plants, and they were able to isolate a few of these manganese oxidizing bacteria. Unfortunately, they didn't have the time to fully characterize these organisms, but that's something that we will perhaps keep looking into. The interesting thing, so I said these manganese oxidizers, they're not usually linked to energy conservation when they oxidize manganese, but another really interesting physiology that was found in drinking water just last year, a group in California discovered manganese oxidizers that do link manganese oxidation to energy conservation and growth. So that would be something really interesting to look for in drinking water biofilters as well. So another cool microbial physiology, new microbial physiology discovered in tap water. All right, and then if we move on to the organic carbon oxidizing bacteria, so the most important thing that we were actually looking at, that we wanted to look at, was the potential for the removal of xenobiotic compounds. So shortly before I left, there was a report that came out saying that 40% of the Danish groundwater wells actually are contaminated with pesticides. And if you remember, this was something that was, well, the use of pesticides is regulated by the drinking water treatment process in Denmark. So this is concerning because they may have to increase the treatment intensity of their process. And so about 11% of these wells have levels of pesticides that are above the regulatory limits. So what we wanted to know is if the bacteria in the drinking water filters actually have the potential to degrade the xenobiotics. And so we actually did find a pretty high abundance of pathways for the biodegradation of xenobiotic compounds in all of the metagenomes of the communities for the drinking water treatment plants. And so these were the main pathways that we found. So we found pathways for the degradation of things like atrazine, phthalates, dioxins, hydrocarbons, and caprolactam. So actually the drinking water treatment filters may be able to deal with this pesticide contamination by actually having biological oxidation of these compounds in the drinking water filters. Whether this will actually occur in situ is something that's still being investigated by a number of researchers in Denmark. All right, and then the last thing that we looked at was we wanted to look at what happens to the microbial community as these cells flow through the filters. And so I had a master's student, Hannah Erland, who was looking at the number of cells that were present both in the sand, but then also in the water. So she had a few different goals, but it was really interesting to us to see that the number of cells, so this is the number of cells times 10 to the 3 per mil in the water. Across the filtration process, a lot of the cells are removed. So after the primary filter, most of the cells are removed. After the secondary filter, there are still some more additional removal of some of the cells. And so something that we didn't quite get to was if the communities in the groundwater really reflect the communities in the final water. We didn't get to look at that, but other researchers have actually found that the drinking water is actually the drinking water microbial community is really determined by the filter communities. And so we had a good idea of what the filter communities were, and we think that that's the drinking water microbiome that occurs. All right, so just some conclusions and then future directions for this work. So in groundwater fed drinking water filters, the role of Chromox nitrospira is quite important. This is especially if nitrification problems are occurring, which is one of the most common problems that can occur in these filters. And we developed a new assay for detecting and quantifying these Chromox nitrospira. The biological functions that we found in the filters were greater than just nitrification, however. So we found manganese oxidizers. I didn't talk about them here, but we also identified iron oxidizers. So one of my colleagues identified and isolated iron oxidizers. There's a potential for methane and sulfide oxidation despite the extremely low concentrations that we see. And there's also the potential for xenobiotic oxidation. And the last thing is that the communities in these filters feed the downstream distribution system. And this is something to consider in terms of the drinking water distribution system that you're looking at and the treatment process that you're looking at. So one thing that we're wondering now is in these drinking water systems where the water that's distributed is biologically stable, what is the impact of that drinking water microbiome? So there would be a major difference between a disinfected and a non-disinfected system. So this native drinking water microbiome exists in biologically stable water, and it's not changed or shouldn't be changed much over the distribution system because there is no disinfection step. So this native drinking water microbiome exists. We know that it's there. It may play an important role in preventing the invasion of pathogens later on through the drinking water distribution system. In the cases where you have disinfection at the end of a treatment train, this results in the reduction in cell counts, and it also results in changes in the community composition in the drinking water microbiome. So there is still a drinking water microbiome, and in fact, it's often is still the same size or a similar size, around 10 to the 5 cells per mil, is sort of a typical range that you see in both disinfected and non-disinfected systems. But because you're really changing this community by doing this disinfection process, you might be allowing for the regrowth of opportunistic pathogens. And some of these opportunistic pathogens, it's been shown that they actually develop resistance to residual disinfectants like chlorine. Another thing to worry about is the negative health effects of disinfection byproducts. So one thing that we're wondering now is if the native and non-pathogenic drinking water microbiome actually prevent the regrowth of putative pathogens? And also, what are their impacts on the human microbiome, since we're drinking probably a couple of liters of this a day, and we're also showering in it and using it for everything. So what is the impact of this drinking water microbiome on the human microbiome? And what is the impact of this on human health? And so with that, I would like to say thank you to you for listening. And I'd like to acknowledge some of the people that did a lot of this work with me. So my colleagues from the Technical University of Denmark, Alex Palomo, Arda Goulet, Florian Wagner, and Bart Smets, as well as the people who funded this research previously, the Willem Fonen and the Marie Curie Actions. And then the people who are continuing to fund my research here at SFU, NSERC, and CFI. So thanks for listening, and I'm happy to take any questions.